And so that kind of finishes my talk about antibody drug conjugates. And now I'm going to discuss bispecific antibodies. So it's again the same antibodies, the same Y-shaped protein, the same monoclonality, so produced in a lab in the mice. But now there is a word two. So bi means two. So we have a word like bilingual, meaning knowing two languages. Bicycle, meaning having two wheels. So bispecific antibody means that there's two parts with that antibody. So going back to our, structure of the Y-shaped protein structure of antibody. So you can see that Y-shaped structure has two ends, one on the left, that's I circled in the red, one on the right. And you can see that those ends are identical. So when they're gonna bind the same green antigen that I showed you in the beginning, so that means that this antibody is monospecific, meaning it only can attach one thing. But you can actually design an antibody where two different parts of that Y structure, one part is going to attach to one thing, another part is going to attach to another thing. So that's shown on the right side of the slide. So it fits two different puzzle pieces, and that's why left is in green, right is in brown. And then you can pretty much take the traditional antibody, that Y-shaped protein that have an identical ends, and then you can do mix and match. And you can design all kind of stuff. As you can see here on the right side of the slide, you can have, there is a green parts and a yellow and the blue parts. You can actually do what's called a tetrabody, where you actually have 4 connections. And so we do it all in the lab. And the idea is that that bispecific antibody is going to bring two different things together. So what two different things can bispecific antibody bring or bridge? So they can bridge cells. They can take an immune cell and just forcefully force the immune cell to go next to the cancer cell. So it kills the cancer cells rather than them just floating around. You can bridge, can take 2 tumor cells and bridge them together. And by doing that, you scramble the signal of that tumor cell. You can bridge receptors. You can take a tumor cell and then take, let's say, a receptor again on an immune cell on some other cell and just bring them together. Or you can bring the cytokines. So cytokines are the structures or the molecules that has an ability to kill things. And so you can also bridge the cytokine. So this is kind of a cartoon example. So you have a bispecific antibody, again, that Y-shaped protein that I've been talking about, that airplane. And it basically, what it does, it takes a part of the, like here on the left side, it's an immune cell, the T cell, NK cells, and it basically forcefully attaches to the cancer cell. And then on the very right part of the slide, it's showing you how they can attach different cytokines and also help that to kill cancer cells.